microphone working? I think so. Okay, perfect. So good morning, everyone, and thank you very much for coming to our meeting in the Fertility, the maternal inheritance. And the first session, actually, we have three speakers, like Jasmin and from ETH Zurich, and Katsuhiko from Osaka University, and Germline from Collège de France. And they will basically tell us their stories. So I just want to give a brief introduction for everyone and don't want to make this longer. And so I think we will start the first talk, like Jasmin is going to tell us about dormancy in hematopoietic stem cells. Right? Thank you.
Thanks a lot. So my name is Jasmin Rettkowski. I am postdoc in the lab of Nina Kabezas-Waldscheid at ETH Zurich in Switzerland. And first of all, I would like to thank for giving us the opportunity to present our research here at the conference in Paris. Today, I will talk a bit more about hematopoietic stem cell dormancy and how we can use this concept of dormancy for therapeutic use. So we all know that dormancy is a very fundamental mechanism that is shared by many, many long-lived cell types, from oocytes to over-muscle stem cells to neural stem cells. And today I will take you on a slightly different perspective, looking at the dormancy of hematopoietic stem cells, or short HSCs. So in our lab, we are interested in how this dormant state in HSCs is regulated, how it's maintained, during steady state, during aging, and how it responds to external cues such as diets or stress. And to study this, we make use of a very broad mix, from dietary intervention models to functional analysis. We use a lot of single-cell multi-omic approaches, and all our methods are also optimized for low input. So for example, we have established several metabolomic state-of-the-art tools using as few as 5,000 cells. And this includes targeted metabolomics, untargeted metabolomics to characterize the molecular and metabolomic fingerprint in mouse and also human HSCs. Because the ultimate goal is, of course, always to translate our findings to human. And today I will share some of our findings that we have gained so far. But let's start having a look at the system we are dealing with, which is the hematopoietic system. The hematopoietic system resides primarily in our bone marrow, where it gives rise to billions of blood cells every day. And as you can see, the system is hierarchically organized, having the so-called hematopoietic stem cell, or short HSC, sitting at the very top. And those cells have the unique capability to differentiate over several multipotent progenitor populations into fully differentiated blood cells. And our lab focuses on those HSCs that actually present the smallest cell population in our bone marrow, and they are characterized by the highest cell figure and the highest quiescence in the bone marrow. And what makes these cells also quite remarkable, in my opinion, is the capability to switch between a dormant and an active state. But what does dormancy mean in HSC? So these dormant HSCs, they are characterized by two key features. First of all, they are highly quiescent, residing in the G0 phase. And we think that around 20% of all phenotypic defined HSCs are only dormant. And second, these dormant HSCs, they are the most potent stem cells in our bone marrow. And the potency of, or better to say, the self-renewal capability of HSCs is usually assessed in transplantation assays, when you transplant these HSCs into conditioned mice and assess the capability to reconstitute the entire blood system. And these days, we have also come to understand that HSC dormancy is not an on-off switch, but rather a continuum. So these HSCs, they can dynamically shift from dormancy to activation in response to cues, and this shift towards activation goes along with almost any kind of increase in their biosynthetic activity. And what is also quite important, that the concept of dormancy is not restricted to mice, but it's also applicable to humans. So we and others have shown that also dormant HSCs reside in the human bone marrow. Of course, not accessible by transplantation assays, but here a couple of surface markers have been identified that label dormant human HSCs. So why is dormancy very critical in HSCs? Dormant HSCs are kind of the guardians of hematopoiesis because every day we are facing challenges such as viral or bacterial infections. And in order to cope with those challenges, HSCs need to enter this activated state because only in this activated state they can give rise to these differentiated cells that can cope with those challenges. And luckily, these active HSCs, they are also able to bounce back to dormancy. However, what has been shown is that a serial exit of this dormancy is quite detrimental because this might provoke DNA damage, genomic instability, and on the long run, it can also lead to loss of HSC functionality, meaning that preserving this dormant state of HSCs is actually key in order to maintain a healthy stem cell pool during our lifetimes. And even though the hematopoietic system resides primarily in the bone marrow, when we zoom out, it's known to have a broad impact on multiple organs. And one of our projects dealt with the crosstalk between the heart and the bone marrow in the context of myocardial infarction, or short MI. So you might wonder why MI? So first, statistics tell us that 1 out of 10 individuals, they will suffer cardiovascular disease. And 50% of them, they will experience MI. So we have an extreme high incidence of MI. The good news is that due to advances in clinical treatments, the mortality rate of MI has actually continued to decrease over the last years. But the downside is that now we are facing a new challenge, which is an increasing number of these patients developing heart failure. So why is that happening? Well, these current guidelines, they are very excellent at addressing this acute life-threatening aspect of MI, which is the block of the coronary artery, but they oversee the long-term consequences, which is the development of scar, the fibrotic tissue at the expense of the heart function. And this is basically what we call heart failure. So basically these days you will survive MI most likely, but you are left with no regenerative therapy. And this is where this project jumped in because it seems like our own immune response is a major player in driving these long-term consequences. It has been suggested that as a response to MI, in mice, HSCs get activated. They give rise to a lot of pro-inflammatory myeloid cells, and these cells exit the bone marrow and start to infiltrate the infarcted lesion in order to provoke recovery and healing. However, what has been shown is that an excessive production of these inflammatory cells is rather detrimental because this leads to excessive inflammation, excessive scar formation of the heart. And the heart is a very flexible muscle that pumps all the time, and basically the scar formation stiffens the myocardium. That limits the contractility. And this is what we call heart failure. And this concept has been partially suggested in mice, but not in humans at all. And this brings us to the first question of this project, which was if MI also leads to human HSC activation. And to tackle this, we teamed up with our bioinformatician in the team that I want to highlight here, Mari-Carmen Romero-Molero, and also with a cardiologist in Freiburg in Germany, who brought all the cardiac expertise to the table. So to address our question, we started to collect more than 150 bone marrow samples from the sternum of patients during open heart surgery. And these patients have been stratified into control patients having no history of MI and MI patients with a clear history of myocardial infarction. And then we performed a single-cell RNA-seq on these hematopoietic stem and progenitor cells that we isolated from the sternum. And here in red, you can appreciate the more inactive, the more quiescent HSCs, and in green, the more active, more cycling downstream multipotent progenitors, the MPPs. And what we have actually observed is that upon MI, there is a shift of this more quiescent, more dormant HSC pool towards more cycling, more activated MPPs. And also in gene set enrichment analysis, in line with that, we observed that these MI HSCs, they really tend to downregulate these dormancy, quiescent signatures while upregulating more progenitor-like, more activated signatures. Functionally, what we did is we transplanted then these MI HSCs into humanized mice, which is the gold standard to assess the self-renewal. And here we observed that indeed these MI HSCs also functionally, they perform worse over time. So I hope I've managed to convince you that yes, human HSCs are indeed impaired upon MI. And this naturally leads to the next question, because then we wanted to know, okay, are these HSCs the true contributors of these myeloid pro-inflammatory cells that are so detrimental in driving heart failure? And actually, this is under intense debate in the field, so whether HSCs give rise to downstream progenitors in emergency situations, or if rather some downstream population takes over this job in emergency hematopoiesis. But to tackle this, we teamed up with Alejo Rodriguez Fraticelli in Barcelona, and we made use of a lineage tracing mouse model. This is an FGD5 specific model, so very specific for HSCs, and once induced, these HSCs, they turn D-tomato positive, and this D-tomato label is then passed on to the progeny, which allows tracing. And we wanted to know if we can detect D-tomato positive cells at all after MI, and if so, if they really make their way and infiltrate the heart. So to test this, we subjected these mice to MI by performing left coronary artery ligation. These baseline mice, they are control, undergoing no surgery. They are only induced. The sham control undergoes only a mock surgery, and MI undergoes the ischemic condition by performing LAD. And what we have observed applying a mathematical mixed model is that indeed the HSC compartment gave significantly rise to downstream cells upon MI, even to more myeloid-committed progenitors. And indeed, these myeloid cells, they really made their way and infiltrated the heart. So these are myocardial sections where you can appreciate here in red the D-tomato, the HSC-derived cells accumulating in the infarct. And to further understand, of course, where these cells go within the heart exactly, we also analyzed those stainings, separating them to remote, not affected upon MI, border, separating infarct from border zone, from remote zone, and the infarct zone. And the infarct zone is where the ischemic conditions kick in. And basically, we observed that indeed these HSC-derived cells, they significantly accumulate in the infarct zone, meaning that HSC-derived cells, they specifically target the most damaged area of the heart upon MI. But of course, we wanted to understand what these cells are exactly, so we also performed a flow cytometry on them. And here we found them to be mostly of pro-inflammatory origin, especially the majority was the Ly6E high monocytes, so the highly inflammatory monocytes. To characterize them also in more detail, we then single sequenced these cardiac myeloid cells infiltrating the heart. And here we identified one cluster that was characterized by a particularly high inflammatory response, here in red, containing all these pro-inflammatory monocytes and pro-inflammatory neutrophils. And then when annotating our HSC-derived cells, we found them almost exclusively present in this highly inflammatory cluster. And this is why we also believe that HSCs, they are not only contributors, but most likely they also drive much of this inflammatory response in the heart upon MI. So using this knowledge, we wanted to know, okay, is it now somehow possible to modulate the activity of HSCs, to target the dormancy of HSCs in order to prevent this myeloid production and improve the cardiac healing thereby? And to identify potential modulators, we dig deeper into the molecular changes happening in these human MI HSCs. And here we found that human HSCs, as a response to MI, they significantly downregulate the retinoic acid, so the vitamin A signaling. And this immediately caught our attention because we have also published an alternative retinoic acid pathway present in HSCs. So we know actually that these vitamin A metabolites in HSCs, they protect HSCs from getting activated upon stress. So, of course, it was very tempting to assume that maybe these vitamin A metabolites, they do have the potential to target HSC activation, reduce their activation, reduce this myeloid detrimental production, and thereby improve the cardiac healing on the long term. But of course, first, to test the potential of these vitamin A metabolites in a human context, what we did is we isolated healthy human HSCs and we treated them with the two main metabolic active forms of vitamin A, which is ATRA and 4-oxoretinoic acid. And when treating those human HSCs with those metabolites, we indeed observed that we preserve a more dormant, a more quiescent inactive signature when sequencing them upon culture. In functional assays, So for example, single HSC division assay, where we sort single HSCs into individual wells and then we trace their division after 48 hours as an indicator of their activation. We observed that these metabolites made them to proliferate significantly less in culture. And also functionally, we performed colony-forming unit assays. So here we sort certain number of HSCs into very thick media that contains growth factors and push cells into the formation of colonies. And the higher the number of colonies, the higher the self-renewal capability. And as you can see, these vitamin A metabolites, they even preserved their self-renewal capability upon culture. So yes, these vitamin A metabolites, they do have the potential to modulate also human HSCs. But of course, to further investigate their potential in the context of MI, we made use of a mouse model. So here we subjected mice to MI followed by ATRA treatment. So for everyone who's not familiar with ATRA, it's already a clinically approved drug. So we thought, okay, why not to make use of its clinical availability? So we subject these mice to MI. We have a sham control undergoing only a mock surgery. We have the MI group that underwent LAD by control treatment. And we have LAD followed by the ATRA treatment. And it made us very happy to see that indeed ATRA is able to reduce transcriptionally the activation of these HSCs, so we maintain a more dormant signature compared to control treatment. And also functionally, our data showed that ATRA dampened this MI-induced increase in proliferation of HSCs. And this also had downstream consequences. So as a consequence, we observed reduced numbers of myeloid cells being produced in the bone marrow, and also fewer cells consequently infiltrated the heart. And this is considered as being very beneficial for the healing. However, when we checked the hearts themselves, we were quite disappointed, because here we observed at later stages of the healing an accumulation of these myeloid cells in the remote zone. And this is actually linked to worsened outcomes. And indeed, when checking the cardiac function by echocardiography, there was no improvement. So while ATRA really protected HSC dormancy after MI, it also led to some functional cardiac side effects locally on the heart. And of course, we wanted to know, is there any way we can circumvent these functional cardiac defects? And to address this, I would like to dig a little bit deeper into the world of vitamin A signaling, because the classical retinoic acid pathway has been indeed described as having a pro-differentiation, even a pro-inflammatory role via the retinoic acid receptor Ra-alpha. So when checking this retinoic acid expression in the heart by bulk RNA-seq, we indeed observe that these cardiac monocytes, they do express extreme high levels of retinoic acid receptor Ra-alpha, and this might explain why they respond in such detrimental way. However, as I mentioned, we have also published this alternative retinoic acid pathway present in HSCs. So we know that in HSCs, ATRA is actually metabolized to 4-oxoretinoic acid, and it's specifically for oxoretinoic acid that binds to Ra-beta, not Ra-alpha, in order to maintain HSC quiescence and dormancy. So when now going back to our retinoic acid receptor expression in the heart, we also observe that Ra-beta is not really expressed here. And this led us explore an alternative approach for oxoretinoic acid. So we thought that by using 4-oxoretinoic acid, we can more specifically target HSC dormancy without affecting this cardiac tissue locally. So to test our hypothesis, we subjected mice to MI followed by 4-oxo treatment. And very similarly to ATRA, also 4-oxo was able to dampen the complete differentiation trajectory of these HSCs upon myocardial infarction. And this dampening effect on HSCs was significantly, particularly present in our single-cell RNA-seq data. Here we observed that upon MI, there's a shift of this HSC compartment towards more cycling, more activated MPPs, very similar to the human data that I've showed you in the beginning. However, it was exactly that shift that was dampened when you give 4-oxo to the mice after MI. And usually after MI, we know that scar formation dominates, but as you can see, 4-oxo was remarkably efficient in inhibiting the scar formation in the downstream cascade, so much that it even improved the cardiac function in these mice, preventing heart failure. And this made us very happy to see, actually. And I just want to mention here, mechanistically, we also, of course, performed the same experiments in raw beta knockout mice where FOXO did not mediate any beneficial effects. So we really believe that FOXO relies on the transcription factor raw beta to promote these beneficial effects on HSCs. And then there was always the final, last question: how translatable are your findings to human now? And to address this, what we did is we isolated human MI HSCs, so HSCs that are already impaired upon MI, and we treated them ex vivo with FOXO in order to check if we can maybe counteract or rescue them. And indeed, when you treat them ex vivo with FOXO, you counteract these functional loss, so you preserve again a more dormant and more quiescent-like signature upon culture, and also functionally you dampen this increased proliferation by MI. Mechanistically, what we did here is we then started to compare the differentially expressed genes, the DEGs, of retinoic acid-treated mouse HSCs with retinoic acid-treated human HSCs. And here we also found a significant overlap of upregulated genes shared by mouse and human and linked to HSC quiescent and dormancy, and at the same time a significant overlap of downregulated genes shared by mouse and human and rather associated with cell cycle and activation. And actually also the primary retinoic acid target genes that we have previously identified in mice turns out that they are significantly enriched in our human data set. And this is why we believe that these retinoic acid signaling might be at least partially conserved among mouse and human HSCs. And this is already the summary of the story. So we have seen today that human HSCs are impaired upon MI, transcriptionally, functionally, and this impairment might be even propagated to downstream populations such as monocytes. Using lineage tracing, we identified that HSCs are really contributors to this detrimental inflammatory cell production. And using this knowledge, we were able to target and modulate the activity of HSCs. We prevented this myeloid detrimental cell production, the inflammation in the heart, and thereby we improved the cardiac function. And what we have presented in the story is a very novel concept of modulating the activity of HSCs in order to improve the function of a distal organ. And in this case, it was the heart, but of course, at the moment, we are exploring other inflammatory disorders that are mediated by overactivated HSCs. And with this, I would like to thank, first of all, my whole lab for their very great support, especially my PI, Nina Cabezas-Walscheid, for her amazing support. Also, I want to highlight here Mari-Carmen Romero-Molero, who did all the bioinformatic analysis. So if you have any questions in terms of bioinformatics, I'm more than happy to connect to her. I'm also very grateful for all our collaborators and funding agencies. And of course, I would like to thank you for your attention. And I'm very happy to take questions.
Questions from the audience? Yeah, Maria.
There are some drugs that use the retinoic acid pathway already in the market, like emulsioners with retinoic acid and antibiotics, and could those drugs be used to improve the repair of after cardiac failure?
Thanks for asking. Just to make sure that I got the question correct. So you were wondering about the drugs that have been already approved in terms of retinoic acid and that are in the clinics?
Yes, exactly.
So indeed, Altrans retinoic acid is an approved drug. It's primarily used for leukemia, for treating APL, because it overcomes this differentiation block of promyelocytes. It's also used for inflammatory skin orders, but not used in the context of cardiovascular disease. And indeed, we have shown that the use of Altrans retinoic acid is even quite detrimental because it's very pleiotropic, binding to all retinoic acid receptors, and does induce these local side effects. So actually what we would advise MI patients is to avoid taking Altrans retinoic acid when using. Our specific compound, this retinoic acid, is not in the clinics yet. So it's not even off-label use, it's not approved, but we are at the moment currently working on that to push clinical phase 1.
Can you tell us more about the dormant state of these HSCs? What are they doing in terms of transcription and translation?
So we know that these dormant HSCs, they are highly inactive. So also compared to their active counterpart, they show the lowest translational activity and transcriptional activity. So they induce some transcriptional programs like HOXP5, for example, RA-beta, that maintain HSC quiescence, but biosynthetic-wise, they are very, very inactive, also metabolically very inactive. Only when activated, they increase these activities also transcriptionally. Now, here in this context, mechanistically, transcriptionally, how it works is that we believe retinol is converted to ATRA by ZIP26B1, and 4-oxyretinoic acid activates this transcription factor RA-beta that directly activates not only retinoic acid response elements, but especially inducing these transcriptional programs that enhance quiescence and maintain quiescence. So basically, they counteract the activity.
Quick question related to that. How long can they stay dormant?
So we believe that really these dormant HSCs that make up only the minority of HSCs, they divide as few as 5 times during the lifetime of a mouse. So they are extremely dormant.
So they can stay dormant for the whole life and then activate after a year? Or, I mean, I meant like how long can they stay dormant before they, have to be activated?
I think highly depends also on the exposure of these HSCs. So it has been shown that when you have chronic disease, you will have a consistent inflammatory exposure, and this makes them activated several times, several rounds, leading to the exhaustion of the stem cell pool. Time-wise, the dynamics, I think they are not exactly known how many weeks or months it takes on average for the HSC to be activated and to bounce back to dormancy. But they are not, usually, if they are really dormant, they should not divide that many times during the lifetime.
So I was wondering whether these HSCs, the dormant HSCs, are a homogeneous population, or whether you think that there might be subpopulations that are maybe already primed to become this inflammatory pool, or whether any dormant HSC has the potential to become that population.
It's actually quite interesting because in general the HSC pool can be quite heterogeneous. We know that the minority is dormant. Within the dormant HSC pool, there have been a couple of markers identified, but still it's the shift between dormancy and activation is also a continuum. So it's very hard to identify the most homogeneous HSC population that is dormant. In this case, we made use of FGD5 expressing HSCs that label the top upper compartment of HSCs. So these are homogeneously expressing FGD5. But I guess that we are by far underestimating the effect because there might be also more HSCs that are more heterogeneous.
So one last question maybe?
Did you explore if this whole inflammation situation following the myocardic infarction could activate maybe other dormant cells somehow, the oocyte or the muscle or the neural stem cells?
It's quite interesting. So thanks for asking, for bringing this up. So I have to admit that we did not dig deeper into that perspective, but since it's a very systemic inflammation, because we know that these dying cardiomyocytes, they also release a lot of inflammatory mediators that are in the system, they are circulating. So I guess I would at least, if I would need to speculate, I would assume that many cell types are affected actually. But we never dig deeper specifically into that.
Okay, thank you very much. And our next speaker is Katsuhiko Hayashi from Osaka University. And Katsuhiko is one of the, if not the pioneers of in vitro gametogenesis, and he's going to tell us about his recent work.
Thank you. First of all, I would like to thank the organizers, especially Elvan and Marie-Hélène, for inviting me this very nice meeting and nice city. So I'm very happy to be here and then I'm very honored to be here. So my title is Germ Stem Cells Culture to produce oocytes, but according to the session title, I my talk is going to focus on the dormancy. So actually, the oocyte dormancy is a system to keep to secure the female reproductive lifespan, because the oocytes getting into the meiosis during the gestation time, they no longer proliferative. So instead, they stay dormant and they stay small until the follicular activation, and then being arrested at the protein stage of the meiotic prophase 1. And histologically, they reside in the primordial follicles within the ovarian cortex, and then those oocytes are surrounded by squamous granule cells. We do not know much about the molecular mechanisms, except for the FOXO3, which is very important transcription factor in mice to keep the oocyte dormant. And actually, FOXO3 is regulated by PI3 kinase pathway. Actually, those pathways actually wipe out FOXO3 from the nucleus so that the oocyte get active. And also, TSC pathway is also involved in the oocyte dormancy by inhibiting the mTOR pathway. And those actually insights are supported by the experimental result, which actually FOXO3 or TSC1 knockout actually shows a massive oocyte activation, which resembling to the POI, premature ovary insufficiency. And then clinically speaking, so PI3 kinase or AKT activator actually can induce the oocyte activation, which actually contribute to the giving the mature oocyte into the infertile patients. So all kind of these oocyte dormancy are very important, not only for the biology, but also the clinical aspect. Is mainly to keep the preservation of the preservation of fertility in the infertility patients. So, but our knowledge on the oocyte dormancy is limited, so we really need to gain our understanding of the oocyte dormancy. So our approach is actually the reconstitution. So actually, we have been working on the germ cell reconstitution for almost decades. So which actually produce the oocyte sperm from pluripotent stem cells, and now we are also trying to make the ovarian tissues or testicular tissues from pluripotent stem cells, which actually provide an efficient tool for the better understanding, also an alternative source for the gametes. So far, we could get a mature oocyte from such an ovary organoid made from pluripotent stem cells. In this system, a mature oocyte can be obtained from such an organoid, and then the oocyte can get fertilized and give rise to pups. But this kind of system is not totally equal to the in vivo system. Clearly, there is a difference between the in vivo and in vitro system. There is the primordial follicle, the small oocyte in vivo ovary, whereas the in vitro ovary, they don't have such a primordial follicles. So actually, we can compare this kind of the in vitro in vivo and see what happened, and see what is missing or maybe too much in the in vitro system. So actually, obviously, the missing part is the environment where the primordial follicle can be dormant. So I'm going to talk about exploring the environmental factors and the gene regulatory network governing the oocyte dormancy using reconstitution system. So simply, we compare the gene expression profile between the in vitro and in vivo, and then find that the in vivo specific gene expression trajectory, like shown in red. So you can see in the bottom, there is a gene expression profile that is specific for the in vivo, which actually represent the primordial follicle stage. And then looking at the GO term, there is the very interesting GO term. So extracellular matrix, response to the mechanical stress, and the response to the hypoxia. Taking advantage of the in vitro system, we can add this kind of factor to the in vitro system, so that we see whether those kind of in vitro system get close to the in vivo system. So to make a long story short, this is published data. So hypoxic condition is contribute to the oocyte dormancy. As you see, number of small oocytes increase under the hypoxic condition. So the hypoxic for sure. And also, I'm going to talk about ECM and the mechanical stress because those are tightly connected. So actually, the ECM is the enriched in the cortical region where the dormant oocyte reside. Then actually the primordial follicle is tangled with the collagen fiber. But if you wipe out the collagen fiber by collagenase treatment, the oocyte get active. So as you see in the second line, you see the cytoplasmic localization of FOXO3, which is a sign of the oocyte activation. So then we have a kind of a hypothesis based on the histological analysis, histological observation, that the oocytes are surrounded by squamous granule cells. Then the squamous granule cells should have a very higher surface tension, meaning that the oocytes are surrounded by such a kind of compression the environment. So my hypothesis was that the oocytes are compressed with such a surrounding environment, and that might be a signal for the oocyte dormancy. So to validate this kind of possibility, what we did, we compress this kind of ovarian piece treated with the collagenase. Even this is treated by the collagenase, the FOXO3 is coming back to the nucleus, which means actually oocytes become dormant again. So then, remaining question is whether this kind of mechanical stress have a kind of direct impact on the oocyte per se, or there is kind of the mediator like somatic cells. So what we did, we isolate the oocytes, and that actually in which the FOXO3 is labeled to be visualized, and then put them into the microfluidic system where we can change the pressure and under the microscope so that we can see the FOXO3 localization in the live imaging system. So in the left part, so if you culture the oocytes without any pressure, with the ligand, the FOXO3 is stay in the cytoplasm. But if you give the pressure, this is the most important part. Can you actually click? Can you click the this right one? Thank you. So upon the giving a pressure, you see the FOXO3 quickly coming into the nucleus, which means actually there is direct effect of the pressure in the oocytes. And then actually this is the quantification. So NC ratio means actually if the NC ratio is going up, then the oocyte become more dormant. So actually the NC ratio increase in response to the pressure, strength of pressure. So actually those are really dynamic the effect. So then we also looking into more mechanistic insight why actually such a pressure provided such the oocyte dormancy. So what we found is actually when we stain the oocytes, dormant oocyte with KIT antibody, you see some strange the signal. So KIT is basically expressed on the surface, but there is the cytoplasmic signal, very clearly cytoplasmic signal. If you look back to the previous paper, actually there is a report showing the cytoplasmic faint staining in the small oocyte in the human ovary. And once they get activate, then such a KIT protein is become in the surface, localized in the surface. So based on this observation, we had a kind of the hypothesis that the KIT protein is somehow sequestered inside the cells, so that the dormant oocyte can be insensitive to the ligand. Whereas once they're growing up, then the KIT protein become on the surface, localized on the surface, making the oocyte sensitive to the ligand. So to validate this kind of the possibility, what we did is actually first we try to see whether this kind of cytoplasmic vesicle, let's say endosome, actually formed in response to the mechanical stress. What we did is we stained with the C-kit and then the culture with the pressure in the left part. So actually the volume of the vesicle actually increased in response to the pressure. And then in the right part, you see this is kind of the live imaging analysis to see how much time do they need. actually they form the vesicle, volume of the vesicle quickly increase within the 10 minutes upon giving the pressure. So, and also following 2 experiments, we try to confirm this phenotype. So what we did is we use antibodies labeled by red, and then also the plasmin, which actually labeled the cell membrane, plasma membrane in green. So in the first experiment, in the condition 1, we just culture them. In the condition 2, we gave the pressure after just after giving the first antibody. And then the condition 3, we gave the pressure after labeled with the 2 markers. Then given that the pressure makes such a vesicle, we expect that the color will be different. So this is the case. If you look at the color, you see in the second lane, you only see the red labeled the vesicle inside the cells. And in the bottom, you see the label, the vesicle was labeled with the 2 colors. Similar observation also seen in this experiment using 2 different colors, 2 different antibodies labeled with the different color. So similar to the previous experiment, we gave the pressure at the first period, just after the labeling the 1 antibody, and then also on the bottom, we gave the pressure after labeling with 2 antibodies. Then expected color will be different. And then this is also a case. You see that in the top part, you only see the red label, the vesicle. In the bottom, you see the vesicle labeled with both colors. So meaning that this actually the vesicle is indeed formed in response to pressure, and then actually surface circuit, the signal circuit protein is actually sequestered inside the cells. And then looking at the biochemical phosphorylation level of the circuit, you see actually phosphorylation level is reduced at least at half level upon giving the pressure you see in the left part. And then also we gave the BPB in the right part. So BPB is an inhibitor of P10. Actually, those are the downstream of the circuit protein. See whether those kind of protein can nullify the effect of the pressure. And then indeed, in the bottom in the right the column, you see by giving the pressure and also giving the BPB, those oocytes get active. So meaning that the action of the sites of the pressure is indeed kind of upstream the upstream the P10, which includes the circuit receptor. So now we have a kind of this hypothesis that under the pressure condition, somehow the circuit protein is sequestered inside the cells, so that desensitize the oocyte to the SCF KIT ligand, which actually express surrounding granular cells. But once they activate the circuit coming up to the cell membrane, so that they are more like sensitive to the ligand. So now we know the kind of the condition for getting oocyte dormancy. So one is the hypoxic condition, one is the pressure condition. Then we try to actually refine more so that we can get very long-lived oocytes in the culture system. So because the oocyte is dormant oocyte is known as the kind of cells that have a kind of specific metabolic state. So we try to change the culture conditions, especially like nutrition, including glucose and the FBS. So here is actually the result. This is very complicated, but you see in the right panel. So actually there is some effect of the glucose concentration in the 1,000, 400, 100. You see the kind of, in the lower one is actually the dormant oocytes. So they actually decrease the cell size. But more interestingly, you see between the 10% FBS and 2% FBS. Actually, the decreasing the FBS is more effective than the decreasing the glucose, which probably means that even if there is the glucose inside in the medium, the cell can't actually take the glucose because the FBS is still actually very low. So then we're getting into try to find some kind of factors included in the FBS. So actually measure a pathway to let cells uptake the glucose is of course the insulin signal and the IGF signaling. So we look at the gene expression level of those receptors in the oocyte. Actually, the dormant oocyte have a higher level of this kind of receptor. So they are really sensitive to this kind of signal. So and indeed, if you give the kind of IGF signaling or IGF-2 signaling or even the insulin signal, this is just the culture isolated dormant oocyte, and then see the NC ratio of the FOXO3. If you look at the right panel, you see if you give the IGF, IGF-2 insulin, actually the oocyte tend to get active. So meaning that this kind of signal actually involved in the at least cell level involved in the oocyte activation. So now we have a kind of such a defined culture condition, and see it's dark, but I'm sorry, but so see what happened in culture system. So this is actually a case of using the fetal ovary. Usually those kind of fetal ovary get activated simultaneously and then grow up many oocytes and then the oocyte run out within like 2 months. Whereas in the lower part, you see the alternative culture condition, you see nothing so much different, and then except for a few follicles growing up. And actually this status can be kept like 2 months. And then we change the culture condition to the normal condition that allow the oocyte follicular activation. And I found that only some of the follicles get active. Because I didn't give the FSH, so actually the growing follicles eventually get atresia. But alternatively, some other actually follicles getting up again. So if you look at this kind of, this is the example of the ovary organoid. You see, in this case, we can keep the 3 months at the dormant state, and then change into the active, change into the culture condition for the active, to allow the active follicular activation. And then looking at the left part in the blue, so you see the many kind of the follicles are seen in the in vitro culture system. And this is very resembled to the in vivo system as I showed in the at the beginning. So probably this actually will mimic the in vivo situation, so that we can see what happened really in this follicular activation. And also this kind of system can be extended like 150, so like 5 months. So actually we can extend it like a year as well. But this is very tough for the postdoc because they need to change the medium for every other day. But anyway, so we can get this kind of FOXO3 positive cells even in the 150 days after the culture. So let me wrap up to the environment factors. So low oxide, high pressure contribute to oocyte dormancy. Upon the pressure, the circuit signaling is attenuated, and then the lower serum actually have an impact on the oocyte dormancy. And the rest of my talk, I will show you a bit, very briefly about gene regulatory network. So because FOXO3 is very important for the oocyte dormancy, not only the oocyte, but also other cell type. So, but we don't know much about the kind of downstream of FOXO3. So we are trying to get try to understand the downstream of FOXO3, at least in the oocytes. In this case, we use kind of reconstitution system for the gene regulatory network. So several years ago, we published that the 4 factors, this is the oocyte-specific factors, can transform embryonic stem cells into the oocyte-like cells like this. So this is the oocyte-like cells made from the embryonic stem cell by expressing 4 factors. And then, in addition to 4 factors, we add the FOXO3, active form of FOXO3. and they found that indeed giving the FOXO3 actually make the oocytes small. So then gene expression profile, although I don't show you, but the gene expression profile are close to the dormant oocytes. Using this kind of system, we try to get the gene expression profile between the dormant state and active state. Oh, okay. So we also get the knockouts, FOXO3 knockouts for the control. So FOXO3 knockouts in the bottom, those are kind of active state in the TG and the wild type. Actually, those factors transactivate the FOXO3. Actually, wild type and TG is very similar. So if you look at the gene expression profile, differentiated expression genes upregulated in the knockout, you see the GO term is related to the metabolic state, like glycolysis or fructose metabolism. So also when we look at the uptake of glucose, so actually knockout shows higher rate of the glucose uptake compared to the transgenic. So and also look at the metabolic analysis, metabolome, and actually there is distinctive the metabolic status between the wild type, TG, and knockouts. And the knockouts show in red in the right. So those are distinctive status. And then looking at the enriched annotations, you see the glycolysis, gluconeogenesis is upregulated in the FOXO3 knockouts, meaning that FOXO3 somehow repress those kind of glycolysis. And then looking at the downstream, actually, by looking at the ATAC analysis, so actually we failed almost for 2 years to do the chip analysis of the FOXO3. So please tell me someone who actually have expertise about this. So, but instead we did actually ATAC analysis and then try to find the downstream genes. Now, one of the candidates is MYC, like this. MYC is the downregulated in the FOXO3 transgenic, and knockout is MYC is upregulated. And indeed, the chromatin state is MYC is close in the TG and open the knockouts. And then indeed, if you give the c-MYC inhibitor there, this is still actually ongoing analysis, so this is very preliminary data. But although the number of the oocytes are not so changed, but the size of the oocyte change. You see in the right dots, you see the size of the oocyte decrease upon the addition of the c-MYC inhibitor. So let me wrap up in the second part also. So gene expression, looking at the gene regulatory network, low glucose intake for sleep. Actually, FOXO3 is enhanced. Then actually the glucose uptake is going down, and then the glucose consumption is also down. And then also c-MYC would be one key key downstream effect of FOXO3 to keep oocyte dormant. So this is it. So with this, I'd like to thank all of my lab members and the collaborators, and I'm happy to take any kind of question. Thank you very much.
We have time for questions. Let's start from the back and we can come forward.
Hi, thank you so much. Maybe I missed it, in which case, apologies. But I was wondering, this pressure that keeps the follicles dormant, do you think it's originating by a special connection of the somatic cells that apply pressure to the oocyte, or is it something perhaps coming from the matrix?
I think the best, don't know exactly, but my guess is that those kind of pressure is generated by surrounding granular cells, because surrounding granular cells in primordial follicles is squamous, which actually have a kind of higher tension. And actually also first sign of the follicular activation is the granular cell become cuboidal, which actually decrease the surface tension. So oocyte, inside of that, oocyte can be a bit relaxed. So I think primarily the surrounding granular cells make such a pressure.
Thank you.
Hi, thank you. I was wondering whether your hypothesis could have any implications in aging, and whether perhaps there's already some evidence showing in an aging ovary if there's more endosome formation, for example.
Yeah, so that's a good question actually. So endosome, we don't know much about the endosome the formation, but we know much about the histological feature of the aged ovary. So the aged ovary in both mouse in the human, there is fibrosis that is kind of accelerated, so that the ovary become more stiff, more hard. So actually, when I talk to the clinicians, they sometimes say that even if there is the oocytes, primordial oocyte in the ovary, they are really stay dormant. And then they also say, the clinician also say that the patient have a very stiff, very hard ovary. So I guess that based on this kind of observation, I guess that this is the case. This actually in the hard ovary. So the oocyte feel the pressure, so that they enforcedly kept in the dormant state. So, and indeed there is in treatment of the clinical treatment, they sometimes just cut the ovary. So those actually cutting the ovary promote the follicular activation. So yeah, so that is a relevant evidence.
Hello, did you look at other mechanotransduction pathways in dormant oocyte, like for example the YAP-TAZ pathway? Is it active in dormant oocyte, in primordial oocyte?
Yeah, actually there is a paper, YAP and TAZ pathway actually involved in the follicular activation, but in the let's say the real dormant oocyte, in the very small oocyte, those actually the pathway doesn't seem to be active, and then also when we gave such inhibitors, they actually doesn't, so the dormant oocyte doesn't actually react very well to such inhibitors. And but those kind of the TAZ, YAP-TAZ pathway is have a function relatively early stage of the growing oocyte. That's for sure.
Thanks. If you relieve pressure in the model of KIT internalization, do you see that internalized KIT gets back to the plasma membrane, or that one is degraded and a new KIT would be put in the plasma membrane?
I'm sorry, I made that.
So in the model of in vitro KIT internalization, so when you apply pressure, if you release the pressure, do you see the internalized KIT coming back to the plasma membrane?
Yeah.
Okay.
So that is kind of reversible. Actually, when we change the pressure while observing the microscope, if they give pressure, those kind of KIT is inside of the nucleus, inside of the cytoplasm, and if you release the pressure, then become like in the membrane. And those are kind of the reversible. But the kinetics seems to be different. The going to the cell membrane is quicker than the coming back to the cytoplasm.
I had a question a bit related to the one from Gabriele. So I didn't quite get what was your hypothesis of KIT going into the endosomal membrane. What do you think is happening then, when you think it's for trapping it, or I didn't quite get the thing.
After forming the endosome, you mean?
Yeah.
So we haven't seen the detail of the fate of such an endosome, but we haven't seen so much actually lysosome, like LC3 signal. So maybe they just stay there and then just circulate, I don't know, but circulating the maybe shuttle between the membrane and the cytoplasm, but we don't have a clear answer actually. I'm sorry.
Hello, I have also a mechanical related question. So when you differentiate the embryonic stem cells into the oocyte-like cells, you need to do reconstitution with the fetal gonad cells, right? So maybe not very practical as a source of potential cells, because you still need the fetal cells. But do you think you need those cells to do the mechanical environment, or are there some other functions? So like, can you omit the fetal gonad step by just making a scaffold that is, stiff enough to allow cells to...
Let me see, that's interesting. We haven't used the kind of the replacements, but let me see. No, we haven't actually tested it. But the interesting thing maybe is that if you change the stage of the fetal ovary, so actually if we use like a 12.5, the most of the oocytes get active. And if you use like a 18.5, many of the oocytes actually become dormant. So probably there is the different kind of cell population, which is actually published somewhere else. And then also there is kind of different feature of those cells. But directly answer to you, we haven't actually tested kind of such a replacement. So that is kind of the
I also have a question to follow up. So do you think the c-kit related to endosomes, do you think pressure changes only the c-kit endosome lines, or does it change exocytosis and endocytosis in general?
That's interesting. Could be because that's true. Because the NC ratio to FOXO3 is in proportion to the strength of the pressure. Maybe a kind of rate of the endosome formation also change. That's why probably FOXO3 is dynamic. And then also the in my guess, not only c-kit, but also IGF receptor also enclosed in such an endosome. That might be my idea, but I haven't done such experiment yet.
Okay, here, one more question. Amazing talk. Thank you very much. I wonder if in your organoids you see spatial differences, because both hypoxia and mechanics, I expect to be variable, in the middle versus in the periphery. Do you see an effect?
That's a very nice question. So usually I saw the dormant oocyte in the peripheral part. So probably those are more stiff than the others. So that's also the case in vivo.
Okay, so thank you, Katsuhiko, and it's time for the coffee break.